Okay, I want to welcome everyone to the webinar this morning. First, I'm going to mute everyone so we won't have any background noises. At the end of the talk, we will unmute everyone and we'll have enough time for questions. So this is the third webinar in the 2010 Become an SPR Expert webinar series. Today's talk will be given by Tsafrir Bravman. Tsafrir has joined Bio-Rad in 2004 as the Biological Application Manager of the Bio-Rad R&D site at Haifa, and is currently the General Manager of our site here in Haifa. Today he's going to talk about the study of small molecules, protein kinase interactions using multiplexed SPR. So with that, I will going to pass over to you, Tsafrir, and good luck.
Thank you, Dalia, very much. So as Dalia mentioned, I will be presenting and giving a talk today in this webinar about the study of small molecules or kinase inhibitors binding to different kinases using the ProteOn XPR36 of Bio-Rad. So the topics of this webinar will include first a general and short introduction about the ProteOn XPR36, the SPR array system, few principles about SPR and how it works. Then we're going to present the study that we performed here, which is an attempt to mimic a small-scale screening campaign of two kinases named P38 and ERK2. Next, we're going to show some results obtained by one of our customers at Chugai Pharmaceuticals in Japan, and they performed also an assay, a binding assay of different kinase inhibitors. And last thing, we're going to show some tips and hints about small molecules assays. There are different configurations and considerations to take into account, and how to perform a best experiment and how to obtain good results. I would like to start the first topic, general introduction, what is SPR, and some introduction to the ProteOn XPR36. So this is the view, the front view of the nice ProteOn. The ProteOn is an SPR-based instrument, and SPR instrument allows you to measure the binding of biomolecules in real time, not an endpoint measurement, but in real time, and without the need of labeling any of the binding partners. So just one quick slide. What is SPR? SPR stands for Surface Plasmon Resonance. It's an optical phenomenon discovered a long time ago in the academy, I think. And this is actually the detection unit. So SPR actually measures changes in refractive index over a surface. It needs to be a gold surface. And these changes in refractive index are directly proportional to mass concentration changes over the chip. So if you have partner 1 and you want to measure the binding of partner 2 to it, so it will generate a change. The binding event will generate a change in the refractive index, which will be proportional to mass concentration changes over the surface. And why do we recommend with SPR? Why is it good? Because as we said, it's label-free. You don't need to label any of your molecules. It saves time. It saves money. It saves disappointment from other reasons. And because it happens in real time, you can measure kinetic, the real kinetic binding event, and not just the affinity. So you can extract the association and dissociation rate constant that characterize the interaction. What are the types of data analysis that you could do with SPR? So the first very basic one is yes-no binding, of course. You have two binding partners, two molecules, which you ask yourself, do they bind or not? So you can easily detect if they bind to each other. Then, of course, the main main feature of SPR instrument is to extract the association and dissociation rate constants that are described by the Ka and Kd. And from the ratio of these two, you can derive the affinity constant or the equilibrium constant, but you can also measure it directly in some interaction depending on the magnitude of Ka and Kd. If they are big enough, the reaction will reach equilibrium or steady state, and then you can determine the affinity constant, and then eventually you can compare these two affinities, one from the kinetics and the other from direct equilibrium, and compare them. Compare these two values, which is another good indication of the quality of the results. Another type of data analysis or application that you can do is to determine the active concentration of a molecule. Usually, concentration of protein is measured by absorbance, but that gives you total protein. Some of it might be unfolded, inactive, and because SPR is actually a functional assay, so it can give you information about how much, what is the percentage in my tube of active protein. And this is usually crucial for biopharma companies that manufacture protein therapeutics and others. Another assay that you can do is thermodynamics. So you can measure the binding event at different temperature, and by plotting the correlation, there are several correlations between the binding constants and the temperature. You can extract the free energy, the enthalpy and the entropy, which then actually explain the magnitudes of why is the interaction fast, why is it slow, why has it a slow dissociation. So all those secrets are lying in the thermodynamics. And the types of molecules that you can detect is anything that will change, that will generate a change in refractive index. You are welcome to test. So this could be proteins, protein-protein interactions, peptides, antibodies, small molecules, DNA, and the list goes on with imagination. So I just want to show 2-3 slides and to lead you to the main feature of the ProteOn and the benefits of it. So 1-2 slides before that. So how does it actually work? How does SPR instrument work? So we said we have two binding partners, and for the sake of all of us being using the same terminology, so we call the one that is immobilized on the surface the ligand, and the other one that is flowing over the ligand is called the analyte. So there is always a continuous flow over the surface, either by running buffer, continuous running buffer, or of your analyte sample once you tell the instrument inject. So this is how it will look like. This slide has two parts. The upper part is a cartoon that demonstrates the molecules inside the flow cell, and below you can see this is actually a cartoon showing how the data is being collected in real time. So at the first stage, you immobilize your ligand or the first partner, and then running buffer is flowing. And since the instrument was zeroed or referenced against running buffer, so the signal is still zero. There is no change in the refractive index or in mass. And once the analyte is injected in the flow cell and it interacts with the ligand, and of course you see a response. There is a change in refractive index, which is proportional to mass concentration, changes over the surface, so you see a binding event curve, binding curve. And once the analyte injection is finished, running buffer is back, and the analyte concentration is actually zero, so you can monitor the dissociation. So in this curve, or what we call also sensogram, you can actually see 3 stages. First, the binding itself, and then if the binding, if the Ka, if the association constant and dissociation are high enough or big enough, the values are big enough, the reaction will reach steady state. and it happens when the rate of association equals the rate of the dissociation, so there's actually no change of mass over the surface, and then you reach equilibrium. When the injection ends, there is dissociation, so the analyte dissociates from the ligand. And this curve, or sensogram, is the output of a single analyte concentration. So one analyte concentration at a certain concentration was injected over the ligand, and you have this sensogram. But to extract reliable kinetic binding constants, you need to have several sensograms coming from different analyte injections. Like any assay, you need several data points. So usually what we do, or what is done in the SPR instrument, is to inject different analyte concentrations. And the main thing here is that most instruments available in the market operate in a sequential mode. That is, there is only one tube or one needle collecting one sample at a time. So you will need to inject concentration 1, and afterwards concentration 2, and afterwards concentration 3, and so on. And to make it even more complicated, between the concentration, you need to remove the bound analytes from the ligand. And this is not a trivial task usually, because you need to make sure two things happen. First, that you remove all the bound analytes from the ligand. So it starts from like empty ligand. And the second thing is to maintain the ligand activity. It's very easy to remove, you can just inject petroleum, but all the ligand will be inactive. So you need to find those delicate conditions to remove all the bound analytes from the ligand. So if you need to inject 5 analyte concentrations, that will take a long time, because it's concentration 1, regeneration, concentration 2, regeneration, and so on. And now I'm coming to the core of the ProteOn XPR, and exactly because of these reasons, we talk about why work sequential if you can work in parallel. And here you can see in this slide, it's a cartoon showing, representing the chip surface. And the unique thing in the ProteOn is that it has 6 microfluidic channels that operates in parallel, independent from each other, so that you can inject 6 different solutions. And in the first stage, you can immobilize or print on the chip 6 different ligands in one time. But of course, you have the flexibility. You can immobilize today channel 1 and 2, and next week channel 3, 4, 5, and so on. And of course, you can immobilize different proteins, mutants, DNA. You have the freedom to use all the 6 channels the way you want. And in the next step, after you immobilize the ligand, you are ready to inject the analyte. So you can prepare up to 6 different concentrations of your analyte, and they will be injected in one time, and this is what we call the one-shot kinetics. So in a single injection, you'll get the binding curve of 6 analyte concentrations interacting with 6 different ligands in one time, in one injection. And by that you can get the results. You can get 6 Ka, 6 Kb, 6 affinity constants with a single injection, the one-shot kinetics. So what you see here in this slide, I'm moving my mouse now, so I hope you can follow it. So here the upper panel is ligand number 1 interacting with the 6 analyte concentrations. And then ligand number 2 interacting with the same analyte concentration, and so on and on. And in this example, one channel was left empty for referencing. You always need a reference. A reference is the analyte flowing over the surface where there is no ligand or where there is no interaction. Like any biological assay, you need a reference, or every life science assay. So one channel here in this example is left empty for referencing, but you can use all 6 channels with ligands and still have a very good referencing, which you can see in this slide. In this slide, this is a top view of the chip. demonstrating the flow of samples. So in the first stage, we mobilize the ligand, and just in a schematic description from top to bottom, the 6 different ligands are printed on the chip. In the second step, the analyte is injected perpendicular to the ligand, and you have a matrix of 36 active spots that we image and measure in real time all the time, all of them. But in between those dark spots, those dark blue spots, we are also measuring the spots in between, or what we call interspots. And on those spots, the analyte is flowing, but there is no ligand. So this is perfectly used for referencing, to remove drift if there are bike effects or any other kinds of things that need to be removed that don't belong to the binding itself. So here on the right you can see all those green squares are the interspots, and these interspots are particularly good and important because they are very local. They are in close proximity to the active spot, adjacent to it, and so it's a local referencing. It's a very good referencing, but of course you're always welcome to try a channel reference, the interspot. All data is being collected all the time. So once the data is collected and you go to analyze it, you have this data. You don't need to choose a priori. So all the data is available, and you can analyze your data with several options and decide for you which is best. And the last slide for the introduction is just to show just few things that I could think of about the flexibility of the protein. So it's not just a matrix of 36 spots. The main thing, to my opinion, is the flexibility that it gives you. So in the ligand stage, you can remove, because you have 6 independent channels, you can immobilize 6 different ligands. As we say, it could be mutants, different DNA strands, or whatever. You can also immobilize the same ligand, but at different densities, at different capacities, which is sometimes very important for very high quality kinetics, because you can see the different kinetic constant at different densities, and they should of course be independent. And another example is to immobilize the ligand and the same ligand at 6 different conditions. Of course, the immobilization itself is also a step that is composed of several solutions that need to be injections, and there are several parameters that govern the mobilization level and the activity of the ligand. So you can test different conditions for the mobilization, and we will show a very nice example in a moment. And for the analyte stage, you can do here in the last sentence the traditional one-shot kinetics, that is 6 different concentrations of the same analyte. But you can also do what we will show here, mainly in this presentation, screening assay, in which you inject 6 different compounds but at the same concentration. Or also something in the middle that I wrote here, but everybody is welcome to create his experiment the way he wants. You have the flexibility, and you can always go back from the ligand to the analyte step. And this is very useful for more advanced application, which perhaps we will demonstrate in future webinars. So, results for the first data I want to share with you is the screening campaign. We try to mimic here a small-scale screening campaign of inhibitors against P38 alpha and ERK2. And just before we start, a few words about kinases, general words. Probably some of you know, or all of you know, but just to give the flavor of importance to them. So kinases catalyze the transfer of a phosphate group onto proteins. Those proteins are the substrate of those kinases. And many proteins, about 1/3 of all human proteins, are thought to be substrate of these kinases. So any malfunction or incorrect reaction, enzymatic reaction in these processes can, of course, generate diseases, some are non-pleasant diseases. Not that they are pleasant diseases, but severe diseases. And this make, of course, kinases very important in a pharmaceutical aspect, and of course they are very popular targets for drug discovery in the pharmaceutical industry. And because there are about 500 known kinases encoded by the human genome, so there is a very high demand for selectivity and specificity, because if you find an inhibitor that is not selective, so all your 500 kinases will be blocked, and that's probably not pleasant. And there are already several inhibitors, drugs approved by FDA that are kinase inhibitors against different cancers, and you can find more information in this reference here below. So, some results. So before we started the screening campaign, we had some kind of interaction, a collaboration, a small collaboration with Merck, and in this, the main point I want to show you here is again the power of parallel of the ProteOn, but in the immobilization stage. As I said before, you can immobilize at 6 different conditions your ligand, and the main point is, of course, to maintain its activity. It's very easy to immobilize, but it's tricky to maintain the activity. So in this example, they had the kinase, or it was an ATPase, but okay. And they knew that the binding site is lysine-rich, and of course, amine coupling is performed via lysine groups with primary amine groups that are in lysine residues of the protein. So they suspected that upon immobilization via amine coupling, the active site will be blocked and binding will be abolished. So what we did, we tested also during the immobilization stage, you need to dilute your protein in a pH buffer, in a buffer having a lower pH, a low pH that is lower than the PI, and therefore the protein is positively charged. Never mind the details now. So here we tested 3 different pHs, and for each pH, we pre-incubate the protein with a non-inhibitor. We incubated with a high concentration of the inhibitor. High concentration, I mean much above the affinity constant, and therefore obtaining that all the proteins will be saturated with the inhibitor, so no protein is left unprotected. So for each different pH, we pre-incubated with a non-inhibitor and without. And here you can see the immobilization scheme in the 6 different channels. And then, of course, we tested the activity with another inhibitor, and you can see very nicely, not surprisingly. Here below, you can see the different, the activity of the protein when it was not protected, so activity was abolished, so no binding activity. And here in the upper graph, you can see the different binding curves, very nice binding curves, at the different pHs when the protein was protected, the binding site was protected, and the protein was pre-incubated with inhibitor. Of course, the signal magnitude varies with the different pH, which is expected. But you can see the same binding curve, the same shape, which is a good indication for the data quality. Another indication for data quality, which is also coming from the fact that there is a parallel mode of operation, is at the final stage of the experiment when you analyze the results. So we analyzed those 3 active surfaces, those 3 proteins that were pre-incubated at different pHs, and we did 2 different independent analyses. One time, we forced the fitting algorithm to find 1 Ka and 1 Kb, a single one, or what is usually called the global analysis. So the algorithm is forced to find a single Ka and Kb for all those 3 surfaces. And you can see the results here in the left table. And on the right, we told the algorithm to find for each surface an independent. I mean, they are independent, they don't see each other. And you can see that the results for the grouped analysis for all surfaces is similar, but also similar to the global analysis. And this indicates that the results, that these are two independent analysis sessions, and it's another indication in the success of assessing the quality of the results, that the results are good and you can trust them. And in the lower panel, you can see the residual plot, and this plot actually demonstrates for each data point of the curve, the gap or the distance between the data point and the fit. So when you see such kind of a residual plot, that all dots are distributed evenly around the zero line, so that means this is the arbitrary noise, and there is very small deviation or any artifact, so the results are good. So for the screening campaign itself, some few points to describe the experiment. So we used ERK2 and P38. We mobilized them on a GLH chip, and of course we used this methodology of protecting the binding site. So we incubated it with a known inhibitor, and then of course, we had 296 well plates, commercial well plates of known inhibitors, potential inhibitors for different kinases. So we screened them, and the point in screening is that you have lots of molecules, so you're not performing a full kinetic assay, but just a screening assay. So each compound is injected at one concentration, and the signal generated are then collected and measured, doing a report on analysis that is done by the software of the ProteOn. There is a very nice way to measure the binding level generated from each compound. And the important point here is that you need to normalize those signals by the molecular weight of each compound, because as we said, SPR measured the mass concentration over the surface, and of course a molecule having 1,000 Dalton will generate a 10 times bigger signal than a molecule having 100 Daltons. So you need to eliminate, to remove this influence. So you normalize, you divide the signal level by the molecular weight. And then the next step is to just draw an arbitrary threshold. Each laboratory or each facility has their own criteria to draw the threshold, and all those compounds above the threshold are considered hits, and we perform to them full kinetics, full detailed kinetic analysis to extract the Ka and Kb. So this is the general description of the experiment. And here you can see the immobilization scheme again of the P38 and the ERK2. So again, we mobilize the P38 once just unprotected, and in another channel protected with a known inhibitor, SB20-3580. You can see the immobilization level here. And also for the ERK2, immobilized unprotected, and immobilized when it was pre-incubated before the injection with ATP to protect the binding site. And the next step after immobilization was, of course, to test the activity of those kinases. So here you can see in this slide the activity of P38. So here P38 in the left figure, the P38 was not protected, and you can see, and we tested the binding with the same inhibitor, but also with another known inhibitor, and you can see the binding activity is abolished, no binding activity. But when it was protected in those right plots, you can see very nice activity, and in the table below, you can see the binding constant, the Ka, Kd, and the affinity somewhere in the 30 or 10 nanomolar. And here on the right, you can see the scheme of those molecules, so you can really believe that they are small molecules. The same thing for ERK2. Interestingly, or not, the ERK2 was active when it was protected and also unprotected. The binding curves were very fast, so we did only equilibrium analysis. Here you can see on the right the data for the equilibrium analysis. and the results show that they have a similar affinity when it was protected and unprotected, so we were sure that these two surfaces are qualified and the ligand is active. So then we started the screening campaign, and as I mentioned, we had 296 well plates, about 200 compounds, a little bit less. So the idea, as I mentioned again, is in each injection to inject 6 different molecules, all of them at the same concentration. I think it was 50 micromolar. And here on the left, you can see some of those curves. And here in the gray area, the gray region, this is the place where we measured the binding level, the signal level, which was, and all was done by the software using a report point analysis. And these signals were then divided also by the software by the corresponding molecular weight. And the next step was to plot the normalized signal for the different compounds. So you can see here the distribution of them. Each dot is for a different compound, the signal level normalized by the molecular weight. We just draw here an arbitrary threshold line, the red line that you can see here. Another thing that you can do is to incorporate in the experiment a positive control and a negative control, and then normalize again, in addition, those signals, saying that my positive control is 100%. It's like it's 100%, and then you can have a relative view of your compound. But this is an option. So the next step was we took all those compounds that were considered as hits and performed to them a detailed kinetic analysis. And you can see here for some of the compounds, for P38, most of them had the Dalia is telling that the slide is only now. So here you can see the P38 slide activity for those compounds we decided on hits. And you can see just 4 compounds with representative interactions. Most of them had very fast binding, which usually is typical for low affinity, because 200 compounds, each of them is selected to different kinases. So we did only equilibrium analysis here. And the same for ERK2, those same for compounds. Again, you can see the different binding curves, and in the table below, the affinity. Okay, so to the second example, to the second result, these results were kindly provided by courtesy of Chugai Pharmaceuticals from Mr. Miura from Japan. They have a ProteOn, the ProteOn is located there somewhere in the chain of the drug discovery, and they studied also kinases. They are looking for inhibitors. The general outline of the experiment, they had 5 different kinases and 1 ATPase, serving as a control, you could say. And they were captured on a GLH chip via biopsy. So they immobilized neutroavidin on the chip covalently with amine coupling, and then did very mild bioaccumulation to those enzymes and captured them. The molecules, the analytes that they were testing were ATP and ADP as a control for activity, as a measurement for the enzyme activity. They also tested a very known inhibitor as a positive control, a known inhibitor for kinases. And the main assay here is that they had 2 inhibitors, which they call A and B, of course, for confidential reasons. These inhibitors were synthesized in-house in Chugai Pharmaceuticals, were fully characterized by NMR, mass spec, and everything that is needed. Binding events were measured at 15 Celsius degrees using 1% DMSO to dissolve those compounds. So here in the first slide of the results, you can see, okay, so you can see here in this slide the 6 different proteins. In channel 1, it was the ATPase, and then the 5 different kinases, noted here also again from confidential reasons A, B, C, D, E. So you can see here that all proteins interacted with ADP, except for kinase C, which is a little bit surprising. But they found later on that it is active, although it did not bind ADP. And in this slide that you will see in a second is the same result but with ATP. So you can see again that the binding events are very fast. So they did only equilibrium analysis for those 5 kinases and 4 kinases and 1 ATPase. And in the next step, they tested the known inhibitor Staurosporine. And you can see here in this slide that, as expected, ATPase did not interact with it because it's a specific inhibitor for kinases. What you can also see very nicely is the different binding shapes or different binding kinetics for the different kinases. And you can see the values of the kinetic constant here for each protein. So they have different kinetics. And then eventually the main thing, they tested inhibitor A, and you can see now that inhibitor A had very good binding to kinase B with 8 nanomolar affinity constant, and a little bit to kinase C with lower affinity a little bit. And for inhibitor B, they detected binding for kinase C with affinity constant of 8.4 nanomolar. And here below is the quotation for Mr. Miura, saying that for these 2 inhibitors, the results that they were generated with the ProteOn are in good alignment with other enzymatic assays that they perform in-house. And this is real science or real development because you get similar results from different assays, different techniques, making the result, having more confidence in the results. Just last thing that they did, they wanted to test the repeatability of the assay. So they injected, they performed a one-shot kinetic assay of ADP. They injected 5 or 6 different concentrations of ADP, and they performed this 8 times consecutively and measured the binding for the ATPase and for kinase C. So you can see here in the left the overlay sensogram. Each color is a different concentration, and from each color there are 8 curves because it's 8 times the same injection. And you can see here at the bottom in the red line is for kinase C, so you can see the injection number or the response unit of the highest concentration, which I think was 400 micromolar. So the signal of 400 micromolar at a different injection, 8 injections, so you can see that it's very stable, it's reproducible and repeatable, and the same for the ATPase. And last thing that they did is they measured the Z factor of the assay. The Z factor was introduced 4 times by Zhang, and you can read more, you can have more information in the reference I'm showing here below. This assay was developed for screening assays, for high-throughput screening assays, and the idea, the rationale was to have some kind of a value to tell you if the experiment or the assay is significant, if the results are, how confident can you be with the results. And this value is specific for each assay because it depends on how much ligand is immobilized, how active it is, the molecular weight of the ligand and the analyte. So it is specific for each assay. And here you can see the equation of it, just very with hand waving. You can see there are 4 parameters here. So one is the average of the average signal of the positive control and the average signal of the negative control. Negative control could be just buffer, which will tell you actually the noise of the system. And of course, the standard deviation of these signals. So you make several repetitions of a certain concentration and also buffer injection, and then you want to compare how significant is the difference between the positive control and the negative control. And you use this simple equation to interpret the results. If you get a value of 1, this is ideal, so it can never exceed 1, of course. The bigger the value, the better it is, because if you want to have a bigger value, it means that the average of the positive control is much higher than the negative, and this term becomes very small. And usually it is considered that an assay having a Z factor of 0.5 and above is considered very good, an excellent assay. And below that it's also okay, but you have to be a little bit to inspect the results in a better way. So what they did in Chugai Pharmaceuticals, they had those 5 surfaces of the different kinases, and they injected different ADP injections, several injections, and they measured the Z factor for the different enzymes. And of course, each enzyme has a different immobilization level, so it's a different Z factor. And you can see here the blue spots for the different surfaces. And from this line, they concluded that for a Z value of 0.5, you need an Rmax of 17. So every surface having an Rmax above 17 would be considered excellent. Below that is also good, but it will have a Z factor a little bit less, a little bit lower. And that was it from Japan. And the last part of the talk, I just want to give 3, 4, 5 slides about general consideration for performing a successful small molecule assay. So small molecule assays are screening, actually, screening campaigns performed in pharma companies. Usually they have huge archives containing millions of compounds, libraries of small molecules, and because the number is huge, they do not perform, as we mentioned, full kinetics. So first they do screening. That means they inject a single concentration of each compound, and they collect the signal level, normalize this level by the molecular weight. It also includes controls, as we mentioned before, positive, negative. They have a threshold they set with their own needs, and all those above the hit are then subjected to full kinetics to extract the Kas and Kds. So general consideration to take into account, small molecules of course can be in the size of 100 Dalton and above, but also below. And remember that's the challenge here, because the SPR response is proportional to actually measure changes of mass concentrations over the surface. So a small molecule has a small mass, a low mass, it will generate a small signal. So to overcome it, you need to immobilize a lot of ligands, and therefore having more binding sites, so more small molecules can bound, and the signal will be increased. Another point to consider is to inject from time to time your control molecule, because the ligand over time, if you're testing many samples, an assay can take hours. So your ligand can, during time, lose activity. And if it loses activity, the signal will be lower a little bit. So from time to time, it is recommended to inject a positive control and then plot, like we saw results from Japan, to plot the Rmax as a function of time to see that the activity of the ligand is not diminished. Another very useful thing to do is to include a competitor to the injection, to do a competition assay, and this is mainly important to eliminate false positives. Because you might get a signal, a positive signal, but it's perhaps because the small molecule was bound to somewhere else in the protein, not in the binding site of it, just not specifically to the protein. So if you're doing a competition assay with an inhibitor that you know, it is known that it bounds into the binding site, So if the signal is lowered now, you know that these two compounds compete on the same binding site, and you know that it is a specific inhibitor and it's not a false positive. Here below, I listed a few more aspects to pay attention: the ligand density, carryover, and the use of solvents. Ligand density, as I said before, the ligand density is mainly important. It's not density, it's the activity. And you can predict what would be the activity of the ligand, or actually what would be the signal level for injecting your analyte, because you can calculate if you know the molecular weight of the ligand and the analyte, and you know how much you immobilize, you can have a rational guess of what would be your signal, described by this simple equation. And the next thing to consider is carryover. Carryover is some of the small molecules have hydrophobic nature, so they tend to stick to the tubing and to the needle. And the problem is during time they will be released from the tubing. And here you can see below an example. In the left, it's an analyte injection, and right after that, it's a buffer injection, but you can see a binding event, a binding curve, a lower one. And this is because a little bit of this analyte was released from previous analytes, and it generated a binding response. So it is recommended from time to time to make buffer injections, and they could be of higher DMSO concentration to remove any sticky compounds. Solvents, usually people use DMSO, it's the universal solvent, but the important point here is that you need to use high quality DMSO. We recommend this one, and you also have to treat it right, put it in dark, use only glass material to take out the sample, because the problem is it will also dissolve, and you can see here below, DMSO is not compatible with polycarbonate, so it will dissolve a little bit of it. You won't see it in the tube, but any contaminants there will bind to the surface, and you will think it's a positive signal, but it's actually contamination binding to the surface. So pay attention and take extra care when you're handling DMSO. And of course, as I mentioned before, visual inspection. I mean, I don't always trust the results just by that. Always have a visual inspection. And this example here, you can see, it's an example of the red line stands for the Rmax. This should be the maximum signal level that could be expected. But sometimes the signals are higher because, for many reasons, the analyte can aggregate, it can precipitate, it can bind to the other sites of the protein, not specifically, and it will generate a positive hit. So this is another example how one can have a false positive without knowing it. And by that, I hope I didn't exhaust you all. By that, I would like to finish and say to everybody, thank you. And if you have questions, this is the time. I will pass I will pass the telephone to Dalia. If you have any additional questions, you can always contact Dalia and myself, and you are welcome. So Dalia, thank you very much.
Thank you very much, Tsafrir. So now I'm going to unmute everyone. So take care if you're talking to someone else. And the webinar is open for questions. Anyone who has questions, feel free to ask here. A second, it will update it soon. Okay, now all of you are unmuted. Anyone?
Yes. This is Cécile from V3. Can you hear me?
Yes, please.
I had one question regarding the protection of the active site with inhibitor. You pre-incubate for how long with the inhibitor? Is it really necessary to pre-incubate with the inhibitor?
This is a very good question because, I mean, you could have, if you know the binding constant, because that depends, how long to incubate depends on the Ka and Kd, and this is in solution. So usually we give it enough time, I don't know, if you give it an hour or half an hour, that should be enough. And you're also choosing a tight binder, a very strong binder with low affinity, so it will have a high Ka. So I would recommend, I don't know, 20 minutes or just to be on the safe side. You could do it for less time, but then you are at the risk that some protein will be inactive. I don't know, 20% of it will be inactive.
Yes, because you will...
I don't have a magic number. Sorry?
Yeah, if you choose a compound to dissociate too slowly, you will get the risk to have no binding afterwards.
Yeah, I mean, if I understood right, this is the other side of the question. I mean, how much to incubate, and then after immobilization, how much time to let it, because this compound needs to dissociate. So you also let it dissociate. You can see that the signal is stable enough. It's a very small change because it's a small molecule, but you let it stabilize, and still all the protecting inhibitor is associated completely from the ligand.
Okay, thank you. Anyone else?
Thank you for your answer.
Okay, welcome.
Okay, thank you everybody for participating in this webinar. Thank you again, Tsafrir. We will be posting the recording of this webinar on our box.net, so you can download it and view it later, and we'll send you a link so you can distribute to all of and anyone else who is interested in it. We have one more seminar to go in this year. The next one will be on September 16th. Professor Gideon Schreiber will talk about pushing the ProteOn XPR36 to its limits from 14-14 docking to nanoparticle interactions. So thank you everyone again, and good day.